Hello, and thank you everybody for joining me for this talk on lipid droplets as part of the FASEB conference. Before starting, I would like to thank Jennifer Lippincott-Schwartz and Bob Faris for organizing the meeting and FASEB for also helping to move this to a virtual space. Of course, I'd much prefer to talk with everybody in person and discuss our science. I'm still thankful that this way I get to share what's been going on in our lab and also to talk about what other people have found recently. Before we get started, I would also like to acknowledge that all the work that I'm going to show today is the result of a fantastic partnership between myself and Bob Faris, my longtime scientific partner and collaborator. Today, I will particularly highlight work from our lab done by Mohan Chitrachut, Sui Wu Sui, Hitting All, Chion Chung, and Maria Jose Solarte, and Niklas Meerd. And we extensively collaborate for structural biology with Professor Mao-Fu Liao of Harvard Medical School and Joel Goodman at UT Southwestern, and for our molecular dynamics simulations with Professors Greg Vogt and Jessica Swanson and Si-Young Kim at the University of Chicago and the University of Utah. So the principal and fascinating biological problem is how systems deal with fluctuations in the availability and need to expand metabolic energy. All biological systems really are facing variation or variability in energy intake and the need to expand energy. And as a consequence, have evolved systems to at least temporarily store metabolic energy. And they do that, as shown in this cartoon here, predominantly in the form of lipids. This is shown for a healthy human that has much more lipid stores as, for instance, carbohydrates. And we think this is a consequence of the unique chemical features of triacylglycerol, the main storage lipid that has a glycerol and 3 esterified fatty acyl chains on it that make this molecule extremely hydrophobic. So you all know what happens when you pour oil in water. An emulsion is formed where the oil droplets exclude all the water and thereby allow the very dense packing of hydrophobic carbons. In cells, specialized organelles have evolved to store and house the lipids, and these are called lipid droplets. They're highly unusual and fascinating organelles because they contain a phase, an oil phase, highly hydrophobic, containing triacylglycerols and sterol esters, but also other neutral lipids depending on cell types, that is bounded by a monolayer, not a bilayer membrane like in other organelles, of phospholipids into which specific proteins are embedded. In addition, lipid droplets, because of their important role in metabolism, are tightly associated, for instance, with mitochondria, but also the endoplasmic reticulum. I'm sure we'll hear more about that. The unique physical features of lipid droplets also have made them observable early on because oil and water diffracts light. Early microscopists, such as the pathologist Richard Altman, have discovered lipid droplets here in a frog liver cell where he drew the beautiful lipid droplets. And discovered them in the 1890s. This is the same person who discovered mitochondria and also coined the term a nucleic acid. Now, in multicellular organisms, the storage in fat, the fat storage is often segregated into specific tissue known as adipose tissue. And in adipose tissue, you can see one very large unilocular droplet occupying most of the cell volume, whereas all the other organelles are sort of squeezed to the side of you. Of course, it's important to remember that most other cell types in the human body can store lipid droplets. The intestine, after a meal, forms lipid droplets. The liver can store a lot of lipids. And in conditions of over accumulation of fat, known as hepatic steatosis, becomes really fatty. The heart runs on fatty acids as a fuel, and often lipid droplets can be observed, particularly also in pathology. And the memory epithelium performs a fascinating biological process in which it forms lipid droplets and then secretes them in an unconventional way to form milk. Now, of course, you're all aware, because the tight link between lipid droplets and fat accumulation and nutrition, that particularly under the conditions of overnutrition or obesity, massive amounts of lipid droplets in adipose tissue accumulate, overwhelming this tissue and leading to pathology. What's a little less appreciated is that when adipose tissue is non-functional or it doesn't develop right, a condition known as lipodystrophy, there is many pathological consequences that are very, very similar. In both cases, the idea is that adipose tissue doesn't work normally. Beyond humans, Lipid droplets can be found in many, many different organisms, many different cell types, including bacteria, fly, yeast, or mammalian cells shown here. And a central question in our lab is, how do cells really make lipid droplets? So in this movie generated in our lab years ago by Jian Chung and Michel Bécrivet, you can see that lipid droplets form at the endoplasmic reticulum. And this is consistent with what other people have found. And generally it is thought that when you zoom in here, in the plane of the endoplasmic reticulum, there are enzymes that take their substrates, fatty acyl-CoA and diacylglycerol, and synthesize triacylglycerol in the plane of the membrane. These enzymes, for instance, DGAT1 and 2, are making a triglyceride, and when a particular concentration is exceeded, there's a phase transition where a new droplet is formed that then buds particularly or specifically in the direction of the cytoplasm and forming a new lipid droplet with the help of a number of proteins that help the droplet forming. Now you can see from the simple cartoon that there's a lot of things which either don't make complete sense or which are really hard to understand just looking at this. So conceptually, you can take apart lipid droplet formation in a number of distinct steps. First, triglyceride is made and the initial lens is formed. Secondly, the initial droplet is forming and budding and growing towards the cytoplasm. Sometime during this process, specific proteins are targeted to lipid droplets that then is being allowed to grow and expand. Consistent with the important role of lipid droplets in physiology, many proteins, either in triglyceride synthesis, lipid droplet formation, or lipid droplet proteins are actually linked to metabolic diseases. Many of them, for instance, found mutated in lipodystrophies or a number of other genetic diseases. Now, today I want to particularly talk about how are triglycerides made and what are they good for? How are the initial lipid droplets formed, and how do proteins target to lipid droplets? So for the first part, how triglycerides are made, the pathway was originally discovered by Eugene Kennedy and colleagues at Harvard and is since known as the Kennedy pathway, in which fatty acids are activated with a CoA to form an acyl-CoA, and then esterified to glycerol 3-phosphate, lysophosphatidic acid, then there's a hydroxyl group free, and finally the diacylglycerol is esterified to triglyceride, the hydrophobic molecule that really is stored in lipid droplets. Now each of these steps is catalyzed by enzymes named after the activity here. So for instance, glycerol 3-phosphate acyl transferase, or diacylglycerol acyl transferase. And what's important to note is that in the mammalian genome, many of these activities are encoded by multiple enzymes. And really for this last step here, which is essential for lipid droplet formation, there are two enzymes, DGAT1 and DGAT2, that were cloned by Bob Farisa's lab and also Calgene for DGAT2 now quite some time ago, 20 years ago. And we can see that these proteins share a name, but otherwise are very, very different. They share no sequence homology, and DGAT1 is a polytopic transmembrane domain protein, whereas DGAT2 has only a hairpin-like structure here with most of its mass on the cytosolic face of the membrane, of the ER membrane. The two proteins have been used to target for metabolic disease, and potent inhibitors have been made by many pharmaceutical companies against specifically either DGAT1 or DGAT2. Two proteins also seem to have distinct cell biology. DGAT1 localizes, in this case, in a Drosophila cell exclusively to the endoplasmic reticulum that you can see here, whereas DGAT2 and some of the other isoforms along the pathway also seem to be able to localize around a subset of lipid droplets, as you can see here. We think that DGAT1 and DGAT2 together make the vast majority, if not all, of triglycerides. When Charlie Harris knocked out both enzymes and differentiated mouse embryonic fibroblasts to adipocytes, differentiation occurred. Normally, the cells are there, but they're empty of triglyceride. They do not have any oil, stained here with oil red. An important goal for us is to really understand how these enzymes work at the interface and catalyze triglyceride. And we recently had a breakthrough when Xiu Sui managed to get the molecular structure of the DGAT1 enzyme that forms this beautiful butterfly of a dimer shown here. And he will talk later in the meeting, so I don't want to steal his thunder, and he will explain to you how this protein really works. Now, what is the function really of triglyceride? And I already told you that probably it's storage. So Mohan Chitraju set out to really establish a system that allows for testing this in more detail. And he generated a mouse model in which adipose tissues specifically lacked DGAT2 and DGAT1. And surprisingly, this mouse looks relatively normal. They have adipose tissue, and you can see that wild-type fat adipose tissue swims up, whereas the knockout adipose tissue falls down and is fixative here because it lacks the triglyceride stores. It's empty of triglyceride. And when you put these mice in cold, which is one of the major inducers of energy generation and one of the major draws on energy stores, you can see that as long as they're being fed, they can maintain body temperature. But when you withdraw food and they become dependent on the energy stores in adipose tissue, they quickly, their body temperature plummets to the point where we have to stop the experiment. So indeed, we do need the fat stores as energy reservoirs for the cells and the organism. But what has become increasingly clear is that what these enzymes do is not only generate the storage lipids, but they also remove the precursors, such as fatty acid-CoA, diacylglycerol, and upstream components that otherwise could be bioactive and often lipotoxic. And so consistent with this, when you overexpress DGAT1 in the liver, you get a fatty liver, but no insulin resistance or pathology. You can do this in macrophages and get a similar result. And other groups, such as R. Goldberg, for instance, have shown that overexpression in the heart of DGAT1 increases the triglyceride, but actually cured lipotoxicity. So we think that that the triglyceride synthesis, particularly of DGAT1, not only serves to produce storage lipid, but also to detoxify. So what happens in humans that lack DGAT1? So when we identified such patients together with Harlan Winter and Mark Daly at a Mass General Hospital, we found that they have a congenital severe diarrheal syndrome, particularly protein-losing enteropathy that's fat-dependent. And so what we think happens here is that in humans, when enterocytes in the intestine are exposed to high levels of fatty acids, they need to temporarily detoxify the lipids by forming lipid droplets. And when you do not have the DGAT mechanism, instead you get toxicity. And with that in mind, Nina Klochowski and others have now formulated a low-fat intake diet combined with supplements to provide essential fatty acids that helps treating the patients with these mutations. Now, concomitantly, this also became a problem for the development of DGAT1 inhibitors as drugs to treat metabolic syndrome, because as you can see here in this data from a Novartis trial, as you increase the dose, you get severe diarrheal syndrome as a side effect. And we think that for this reason, probably DGAT1 is not a great target to develop for metabolic disease. That might not be quite the same for DGAT2, where there's also potent inhibitors. And where recently Pfizer has published data that treating human patients with inhibitors for DGAT2 and for the liver severely or very significantly reduces liver triglyceride very, very fast. And so thereby maybe a possibility to use DGAT2 to target metabolic pathological accumulation of fat in the liver. So how are lipid droplets initially formed after the synthesis occurs? And in our lab, we like to take unbiased systematic approaches to really pinpoint which are the key factors contributing to this. And we've performed a number of genetic screens, initially in Drosophila cells and more recently in human macrophages. And this was spearheaded by Lina Kurvila at Yale and then has been picked up by Nicholas Mayrd since. And in this screen, we knocked down systematically every human gene and analyzed the lipid droplet phenotype associated with that with high contrast microscopy. And when we analyzed these data, we found distinct classes of phenotypes associated with the knockdown of specific genes, and one that really stood out is a gene called BSCL2. Now, this is probably familiar to many of you because this gene is mutated in one of the congenital lipodystrophies, was discovered in 2001, and also was found by Rob Young's and Joel Goodman's group in yeast screens to be really important for maintaining normal lipid droplets in yeast. Among the many groups who worked on this, including Pedro Cavalas, Elena Icones, Larry Chen, Huyi Marx, Xavier Prieur, and Justin Rochefort, now we know that this may form a oligomer in yeast. This was initially found by Joel Goodman and localizes to ER junctions, ER lipid droplet junctions in yeast. So when we analyzed the phenotype associated with this impatient fibroblasts, you can see that whereas wild-type fibroblasts have sort of this normal lipid droplets, there's a really ugly appearance of droplets and sapin lipodystrophy patients where there's a few very large droplets and otherwise sort of this snowstorm. And this can be modeled in other systems such as Drosophila cells where there's a few pre-existing droplets, and then normally during incubation with oleate, only a few lipid droplets are made that are growing in an organized way. When you don't have sapin, however, there's this whole snowstorm of these little lipid droplets that form all over the place. And analyzing them by cryotomography shows that the lipid droplets form sort of these small entities that are separated from the ER and what appear to fail to grow in size over time. So how does sapin do that? So first, we were really intrigued by tagging sapin with a GFP, the endogenous molecule, either in human cells or in Drosophila cells. And you can see that it forms foci that migrate in the ER, marked in red here, and are moving rapidly, almost like scanning the ER. So what are these foci? Xu Wu in the lab decided to solve the structure, and he did so by cryo-electron microscopy. And what he found is that particularly off the sapin structure that consists of two transmembrane domains and a luminal domain and short cytoplasmic extensions, the luminal domain forms a well-ordered oligomer with many subunits forming this ring. Within that luminal domain ring, we observe particularly a beta sandwich fold domain and these small hydrophobic helices which form an almost contiguous ring as part of this oligomer. The overall structural features here are conserved because at the same time, Rob Young's lab, together with Yang Yan, solved the structure of the human sapin, and we've since then also solved the structure of yeast, again, Xu Wu with Henning, and in collaboration with Joel Goodman, and they will talk about this later. And what we overall observed that the subunit number varies and some geometrical features vary a little bit, but overall the complex forms a very similar geometry. And we think it sits with hydrophobic helices embedded in the membrane and the hydrophobic transmembrane domains spanning the membrane, forming sort of this cage structure in the ER membrane. And we at that point thought that probably these hydrophobic helices could attract the triglyceride molecules here in the membrane. And one thing that never sat quite right with us is that when you just purify sapin by itself, you cannot find triglyceride with it. Another thing that is surprising and was first noticed by Jion in the lab, Jion Chong, is that there is the segment with the hydrophobic helices is not only conserved in hydrophobic character, but also in sequence. And so if the only role of this was to interact with triglyceride, why would the sequence be conserved? And Jion therefore thought that maybe this protein has a binding partner, a protein binding partner that is interacting with it. And she designed an experiment deleting specifically this segment and ask whether there's any specific interactors. Analyzing the pull downs of these two constructs, she found specifically this protein TMAM159 only interacts with the wild type and not this version of sapin. And she's done many controls to show that it is a specific interactor of sapin dependent on this hydrophobic helix. So what is this? TMAM159, that we since renamed LDAF, is a small 161 amino acid protein first found in murine fatty liver induced by PPARs that had a very high correlation in our screen with sapin. It may be the homologue of the yeast LDO proteins that have been worked on by the group of Pedro Cavallo, Maya Schuldiner, and Maria Bonard. Here's just to show that TMAM159 and BCSL2 correlate very well with each other with all the parameters that we can measure in our high content screen. So we postulated that probably LDAF works with sapin, and I already showed you it interacts with sapin, but for us to believe that this is an important protein in the process, we also think it should associate with lipids localized to sites of the formation of lipid droplets and also be required for their formation. And so to test this, Jaehyun purified the SAPIN LDAF complex in a heterologous system to apparent homogeneity. And what you find here is that they form a very, very large complex where both proteins appear to be present quasi-stoichiometrically. And I already told you, when you purify SAPIN alone from cells, it is not associated with any triglyceride, really. But as soon as you co-purify the complex, you start to see some triglyceride that specifically co-purifies with this complex. And you can push that if you incubate the cells with a little bit of oleate to induce triglyceride synthesis. And we know that it's triglyceride synthesis because we can block that reaction when we add DGAT inhibitors. So we think this complex really contains SAPIN. And when we started analyzing it by cryo-electron microscopy and comparing it with the cryo-EM structure of SAPIN alone, we found these highly curved micelle shapes. And this leads us to think that within this complex, the triglyceride may be in here and really bulging out the micelle that we can purify. By the way, also then vastly complicating the structural analysis of this. So both proteins also appear to co-localize. I've shown you that they interact and they can form foci in the ER. Many of them co-localize. And only at the sites that contain both SAPIN and LDAF1 can you see initial lipid droplets form. Here's 3 examples. All these proteins are endogenously tagged, and where we use perilipin as a marker for lipid droplet formation. And you can see that it forms within the first few seconds here of the forming lipid droplet only in the sites where you have both SAPIN and LDAF. And this is quantitated here, showing that really we see only lipid droplets forming at sites that have both of the proteins, but not one of them or neither of them. Jaehyun then did a cool experiment where she relocalized the LDAF protein and dragging it together with the ER membrane to the surface of the cell right underneath the plasma membrane using a chemical heterodimerizer strategy. And when you do that, first you can see by turf microscopy that right underneath the plasma membrane, you now get a lot of LDAF and that it brings SAPIN along to the plasma membrane, underneath the plasma membrane, again arguing that both proteins tightly interact. And when you do that and then add oleate to the cells, we now see a lot of lipid droplet formation right underneath the plasma membrane, where normally there is very, very few lipid droplets formed. When you have both proteins there, they form. Now, during the formation, something really fascinating happens to the complex. What you can see here is that initially the proteins co-localize, but as lipid droplets form, the complex segregates from each other, and LDAF1 ends up on the surface of lipid droplets. Here you can see sort of the result here, where the ER is labeled in blue, the SAPIN remains at the junction, and LDAF1 gets blown out onto the new lipid droplet. And not surprisingly then, LDAF is really important for lipid droplet formation because if you delete it, you see much fewer lipid droplets that are different in size and shape, and we can also quantitate that. So from this, we conclude that SAPIN and LDAF are key players in lipid droplet formation, and that they are sufficient to localize SAPIN at the sites of lipid droplet formation. And we think that maybe this complex functions by catalyzing phase separation of TG in the ER. So to visualize this, what we think is going on is that the triglyceride is synthesized by DGATs, and once it reaches a critical concentration, it phases out to make a new droplet, oil droplet, in the complex of SAPIN and LDAF that then is allowed to grow and butt towards the cytoplasm. And if you don't have the complex, the triglyceride is still made, but now has to form at ectopic sites. So for me, at least, this is reminiscent of the model that is proposed for another phase transition, the model for protein folding by the chaperonins by Ulrich Hartl and Art Horvich, where chaperonins basically allow proteins to fold in this little cage here by excluding unfavorable alternative interactions with water and other proteins. And in the case of SAPIN, maybe what happens is that this structure here with this many transmembrane domains and lots of protein mass allows the triglyceride to phase transition within it by excluding otherwise unfavorable interactions that would slow down phase transition between the triglycerides and the acyl chains of the phospholipids. Now, of course, we understand that's only a model. Other models are possible. We need more opinion than really sort through this. SAPIN has also been proposed, for instance, as a scaffold protein, or Rob Young, for instance, has proposed it may act in lipid transfer. Looking forward to those discussions. So now to the next step. How do proteins get onto lipid droplets? I already mentioned that droplets are really important components of lipid droplets, and it was a hallmark discovery by Konstantin Dean-Landos and Andy Greenberg now 30 years ago when they identified perilipin-type proteins as specific lipid droplet proteins. And since then, many groups, including ours, have used different proteomic techniques to identify the protein composition of lipid droplets. And essentially, I found that there's many lipid droplet proteins that are specifically there, including enzymes of triglyceride synthesis, of lipolysis, mobilizing the fat again, but also a number of proteins that we really don't understand why they are on droplets, what they do there, and so forth. Now, generally, at least two pathways can be recognized that mediate targeting of proteins to the surface of lipid droplets. In one pathway, proteins from the cytosol bind to lipid droplets directly using often amphipathic helix type motifs. In a second one, proteins are initially inserted into the ER, maybe by an unconventional pathway that Bianca Scholl and Ron Capito have described, and then move to the surface of lipid droplets. We struggled for a number of years with names for this, and I'd like to propose to maybe call this one here cytold for a cytoplasm to lipid droplet, and this ertold for ER to lipid droplets. You can use the chat box to let us know whether you either think these are great, horrible, or whether you have better ideas. Now, in the ertold pathway that GPAT4 and other enzymes appear to use, proteins are initially inserted into the ER. But of course, they don't jump in and out of membranes to get there. So how does this actually work? Florian Wilfling, when he was in the lab, discovered that the proteins not only move to lipid droplets, but also accumulate. Here you can see that during lipid droplet formation, Drosophila cells, the red protein that is initially distributed in the ER accumulates efficiently on lipid droplets over the time course of lipid droplet formation. Now there's complexity in this because, as I showed you, the proteins accumulate on lipid droplets, but they don't appear to do so during the earliest formation. So something prevents it to go on onto droplets early on. But if you just take a small sequence of GPAT4 that is sufficient to mediate targeting, this small sequence can actually go early on. Now, importantly, this protein ends up on the surface of lipid droplets. You can see here this immunogold staining against GPAT4 decorating the surface of droplet, but not the ER that is close by and wrapping the lipid droplet. So how does this work? What Flo found in fluorescence recovery after photobleaching experiments, pioneered by Jennifer Lippincott-Schwartz, is that the protein during the time course of its targeting appears to, when you bleach it, migrate onto the lipid droplets through direct membrane bridges between the ER and the lipid droplet. You can visualize these, they're abundant and can be seen easily as these ribosome-studded structures that are contiguous with the surface of lipid droplets. And here's just a drawing for where the membranes go. So we think that this here provides the pathway for ertold to allow proteins to travel via bridges onto the surface of lipid droplets. But this brings up another really important problem. Just because there's a connection doesn't mean the protein should accumulate here. It should equilibrate between both compartments, and because there's a lot more ER than droplets, most of it should stay back in the ER. So how does this work? So this is the problem that Maria Jesus Olarte tackled. And she started by asking, like, are there any specific sequence feature in the life drop sequence that is sufficient to target droplets that are needed for this? And here's a cool experiment where she essentially kept the amino acid composition the same, but scrambled them all around. And you can see that the protein still inserts into the ER, but now does no longer target, highlighting that specific sequence features are required for targeting. She then tested a number of other alternative models that I don't have time to discuss, and also mutated a number of the key residues. And what she found was that specifically a number of unusual residues positively charged in this otherwise very hydrophobic segment are required to target lipid droplets. You can see when we mutate them, lipid droplet targeting is much less efficient. And in addition, a number of large hydrophobic residues, and particularly tryptophans that are in this hydrophobic segment, are needed, because if you mutate the tryptophans but not some of the other hydrophobic residues, you abolish the accumulation on droplets. So how does this really look like? So we turn to molecular dynamics simulations with the help of Seyoung Kim, Jessica Swanson, and Greg Vogt. And what we found is that the life drop sequence forms a hairpin in the ER membrane, where the positively charged residues anchor this hairpin onto both sides of the membrane and keep the large hydrophobics in an energetically unfavored position in the middle of the membrane. When you go to a monolayer, the large hydrophobics are allowed to swim up to the surface, and umbrella sampling simulations have shown that this makes an energetic contribution, because now these large hydrophobics are in a much more favorable environment on the monolayer than in the bilayer. And what that really means is that it's more favorable for them to be on the monolayer than the bilayer, and we think that this drives to the accumulation on droplets. So specifically, an unfavorable conformation where the large hydrophobics are kept in the bilayer as opposed to allowed to swim up really drive the accumulation onto the lipid droplets. We've since then extended these findings to ALG14 hairpin and found very similar principles to apply. We also were able to, or Maria Jesus was able to analyze a whole number of potential hairpin forming proteins and classify them as lipid droplet targeting or not targeting. And when she analyzed the whole number of physicochemical parameters for these, we found that this sequence features alone are sufficient to segregate the non-targeting from the targeting ones. And again, that particularly the position of the large hydrophobic residues within that segment, hairpin segment, is particularly important. And we think that this is at least one mechanism to explain lipid droplet accumulation of erythroid cargo. How about the cytosol pathway? In this pathway, proteins bind directly from the cytosol. And a major challenge to explain is like, why do they bind here rather than on the ER surface membrane? Rashid Thiam, when he was in the lab, and Colleen Prevost after him, used a system to visualize this, where there's a giant unilamellar vesicle into which a triglyceride lens is incorporated. And you can see, for instance, in this amphipathic helix protein, CCT, it binds specifically to the surface of what mimics the lipid droplet, but much less so to the bilayer membrane. So what is the surface difference between the two? So again, we turned to molecular dynamics simulations, and Maurice Cohen set up a system where he blew apart the bilayer and inserted triglyceride under this and asked what are the consequences to the surface. So if you turn this and look onto the surface, what becomes apparent quite immediately is that the lipid droplet surface has a number of these very large packing defects. They're much larger, much more frequent, and much more persistent, as shown in this quantitation here, than on a bilayer membrane. In fact, we think that the lipid droplet surface may mimic what's found in highly curved bilayer membranes. In the case of the lipid droplet surface, the triglyceride squeezes in between the phospholipids and pushes them apart, thereby generating a packet defect. In the case of highly curved membranes, pulling apart the head groups generates a packing defect. So is this really important for binding? We can test this in biochemical reconstitutions using peptides from lipid droplet binding proteins linked to environmentally sensitive fluorophores that shift both their intensity and emission spectra on binding to the membrane. And you can see here that you can reconstitute binding to lipid droplets with such a system, but actually quite surprisingly and consistent with the notion that is due to packing defects, these proteins will also bind specifically highly curved membranes, very, very small ones. And so the model that has emerged from this for cytosol is that amphipathic helixes read out the surface packing defects, and we've shown that they use large hydrophobic residues to find them, and that then leads to folding into amphipathic helix on the surface. This essentially also makes this process here energetically irreversible. Now, how is specificity achieved? We don't fully understand that yet, but an important contributor, initially discovered by Nora Corey and Rashid Thiam in the lab, was that lipid droplets are very, very crowded, and they are fully saturated with lipid droplet proteins that often have multivalent binding sites. And they crowd out and bump out proteins that are not supposed to bind. A process also important during lipolysis when the droplets shrink overall and some of the proteins can be displaced. Now, this, I think, is sort of what we believe is going on for the cytosol pathway. I already mentioned in the beginning that the lipid droplet protein contains a number of sort of really unexpected players, and we were surprised to see many of them, and there is a lot of open mysteries. I just want to give you one quick story about one such mystery. And so when we analyzed the lipid droplet protein, we found one particular protein that was really surprising to us. So this here is in THP1 macrophages, where we also did our screen. And we analyzed proteins that enrich on lipid droplet over their presence in the lysate. And you see that the genuinely known lipid droplet proteins, such as PLIN2 or AUP1, are highly enriched on lipid droplets. And so is this protein here called MLX. And what was really bizarre about this is that this is a member of a transcription factor family of basic helix, loop helix, leucine zipper proteins that form obligate heterodimers and shuttle between the nucleus and a cytoplasm in response to glucose, and are sort of main regulators of glucose metabolism and lipid metabolism. The best known protein here is a protein called ChRABP. And what was intriguing is here you can see that this protein can localize to lipid droplets in a number of cell types, including hepatocytes or all these other cell types here. It was also found as a lipid droplet protein in James Oldsman's APEX screen. It's a different proteomic technique for high confidence lipid droplet proteins, and was also found by Natalie Kramer and Matthias Munn laboratory in liver lipid droplets. And so intriguingly, this transcription factor contains a short sequence that is sufficient to mediate its binding to lipid droplets. And within that sequence, there is one of these amphipatic helices that, when mutated, basically abolishes binding. And importantly, this was also a hit in our screen and gave a very prominent lipid droplet phenotype. And I don't have time to show you all the data, but a really interesting and intriguing concept emerged here, in which MLX, MLX-IP, and IPL are able to bind lipid droplets. And when they do so, they cannot go into the nucleus to induce gene transcription. It turns out that, of course, many of the targets here are involved in modulating glucose uptake into cells and lipid metabolism. And this provides the cells for a way to respond differentially whether or not they have lipid droplets. If there are lipid droplets in the cell, those transcription factors are sequestered to the surface of the lipid droplet, preventing the induction of some of the glucose metabolism and lipid storage genes that are otherwise may increase even further the number of fatty acids in the cell. And we think this is a fascinating paradigm for how lipid droplet binding of a protein that we would not normally expect there has been used to really integrate cellular physiology with the presence of lipid droplets. Now, with all of this, I want to leave you with sort of the current model, what we have learned based on what I've shown you in the beginning. We now have a better understanding how triglycerides are synthesized by DGAT1 and how the initial lens may be formed. I refer to Shrivu's talk later in the meeting to explain this in more depth. We've begun to understand how SAPIN and LDAF may collaborate to provide the space for the initial lens formation. We've also started to understand how the ERTOL pathway delivers proteins from the ER to lipid droplets during the initial formation, and how specific sequence features provide the energy to allow for accumulation on the surface of lipid droplets. We've also learned how cytol delivers proteins from the cytoplasm to the surface of the lipid droplets, often allowing amphipatic helix proteins such as CCT or perilipin to bind to lipid droplets, and understand the effect of crowding that crowding may have onto the composition of lipid droplets. But when I go through this slide, what is most apparent to me is not the stuff we've learned, but all the stuff that we really don't understand. And I just want to leave you with what I think are some of the most fascinating questions for us to work on now as a field. Is like, we still don't understand at all how DGAT2 works, whether it works the same way, where the triglyceride goes, and what's the enzymatic mechanism there. Are the same mechanisms that are apparently operating for triglyceride also at work for cholesterol ester droplets and other lipid droplets formed by other lipids? What's the molecular function of SAPIN and LDAF really? Is it just as phase separation? Do they have additional biochemical function? Do they have different functions and additional functions later in your lipid droplet formation? I pointed to some additional data. There's beautiful data also from the Econen's lab on later functions of SAPIN. What is the relationship between the initial lipid droplet forming and the sapin and some of the bridges that must be necessary to mediate protein targeting later. How are these bridges built, I think, is also a fascinating question. And then something that I didn't touch on, but which is prevalent throughout the literature, is that there are proteins that are only found on a subset of lipid droplets, but not others. And right now, we do not know which pathways gets them there and how this distinction is being made molecularly. So I think there is a lot to work on, and I'm really looking forward to continuing this journey. During the talk, I already acknowledged sort of the main contributors from our lab to this, and also a number of the former contributors to this. I also acknowledged, I think, most of our collaborators. I want to highlight Stephanie Moore, Tally Lambert, who helped with a lot of the microscopy aspects, and Shudong Wu and Tom Rappaport for help with structural biology. Sinran and Morven have been a fantastic resource for electron microscopy, and the High Throughput Cell Biology Center at Yale that I directed when I was there started the stream. We had help from Frank DeMeo on the model building for SAPIN and collaborate with Joel and Sean on the structure. And finally, I want to thank particularly the National Institutes of Health and NIGMS and NAGDK for funding this work, as I want to acknowledge Higher Use Medical Institute support, as well as Pfizer, who supports a project in our lab that I didn't have a chance to talk about. And with that, I hope to take any questions you may have, and I'm looking forward to discussing more with you during this meeting. Thank you very much.